Welcome to the video Measuring Biodiversity here in Principles of Ecology 3403. Biodiversity is a relatively new word. It was introduced by Dr. Edward O. Wilson, who joked that although it meant biological diversity, biodiversity was what you took when you combined biology and diversity and took out the word logical. It was not a very funny joke then, and it isn't now. So, what is, he says, collecting, there you go. What is biodiversity? Well, we start with the notion that all of life is a hierarchy. We've learned that in intro bio. Biochemists tend to study atoms through tissues, physiologists cells through organisms, and ecologists focus higher on the hierarchy. Organisms, populations, communities, ecosystems, and the biosphere. Populations are collections of organisms. Communities are collections of species populations. Ecosystems are those communities when embedded into the abiotic environment. And then the biosphere is the whole 9 yards, the whole kit and caboodle, all of life on a given planet. And given this, we can start thinking about biodiversity in a fairly specific way. Well, first of all, your book describes three different ways of doing it, three different ways of counting up the number of species in a given area. One is by taxonomic affinity, that is who the things are related to. So here we have a bunch of warblers, little passerine birds that hang around in trees. Another one is by focusing on what they eat, that is the guilds concept, which in this case refers to pollinators, as a bee, a hummingbird, or a bat. And the third are functional groups, which is more of a kind of a general form of the guild, actually. It's a focus on how organisms are constructed to make a living. So what we have here is an aphid versus a mosquito. They don't eat different things, but they both are small little things. You can squish them pretty easily, and they can inject something into either a human, in the case of the mosquito, or a plant in the case of the aphids. So biodiversity then is the variety of life. It's a measure of the immense variety that we see out there. Now, much of science actually focuses on particulars. How does a muscle cell work? How are genomes transcribed? But biodiversity actually focuses on the other end of the spectrum. Why are things so different? So in a population, why are some critters small versus large? What is the sex ratio of the population, males to females? In a community, it's the number of species or the number of different ways of making living. We just saw a couple different ways of quantifying biodiversity that way. Ecosystems have different nutrient cycles, different kinds of nutrients cycle in different ways. And, well, although we only have one biosphere, some ecologists are very much interested in life in other worlds and what are the conditions that allow that to happen. So there's even a biodiversity among biospheres if you kind of go the Star Trek route. Now, we're going to focus initially on three different ways of quantifying biodiversity. The first is called species richness. It's the number of species that can be found in a given area, the number of grasshoppers in a hectare, the number of bacteria in a gram of soil or a quarter centimeter of soil. Something along those lines. It's simply a number per unit, another number. And this is important because the number of species will vary as you increase or decrease the size of the area in a predictable way, as we'll see. A second one is a little squishier. It's called evenness, and it's the balance of commonness to rarity. And in the textbook, they give the example of a field, community A, in which you have 4 species of mushrooms dominated by a single one that kind of looks, like a little, I don't know what that looks like. Anyway, the second one is a community with the same 4 species. So the species richness is identical between the two, but there's just so much more diversity. They celebrate diversity in this community B, and no single species dominates. Now, we'll use one index to measure evenness, but actually there's another way which I kind of like, and it's called the rank abundance curves. You may have noticed already through your readings that there are a lot of graphs in ecology. As we'll talk about at considerable length, graphs are really the way ecologists talk to each other. They're very economical ways, there's that word again, of describing a pattern. So a rank abundance curve basically describes the balance of commonness and rarity in a community. So on the x-axis, right here, you've got the abundance rank. That is, you take the most common species, that's 1, second most common, third most common, etc. And on the y-axis, you've got their proportional abundance from 0 to 1, if it's a proportion. And this is typically log scaled, log 10 scaled in this case. We'll talk more about log transformations in a while, but you've seen these before. They're kind of warped in an interesting way. So the distance from one order of magnitude, 0 to 0.1, is the same as the next order of magnitude, 0.1 to 1. So let's compare the rank abundance curves of these two fictional communities of fungi. Community A, the one dominated by 1, tells us two things. First of all, there are 4 species because the line stops at 4. And the other is that the abundance varies considerably. The most common thing, moving all the way up and then over to the left, suggests that the most common fungi makes up 0.85, 85% of the community. Moving down the curve, we find that the other 3 species are equally common, another bit of fact, and are represented by 1, 2, 3, 4% of the community. Now let's go on to community B. Same 4 species, line stops at 4, but they're all found at 0.1, 0.2, about 25% each. So in this case, the even community has all the same abundance. The less even community is dominated by single species at 85%. So this is kind of abstract, but actually this is a very common pattern in ecology, which tells you a little bit about how the community is put together. So we'll finish with a simple little example. There was a study of invertebrate communities in Connecticut in 3 streams. One of those streams ran through an urban area, so it got lots of runoff from concrete and such. And other 2 were outside of the urban area, so the stream was less polluted. Some college students basically sampled invertebrates from each of these communities, little creepy crawlies that are found amongst the rocks at the bottom of the stream. And they built rank abundance curves to look at the relationship of diversity and evenness. And this is what they found. So again, we have rank abundance on the x-axis, proportional abundance on the y. In this case, we've broken it down to things that are represent 1/10, 1%, and less than 1/10 of 1% in terms of abundance. And we see 3 very different curves. The first curve, the very steep one, is Centennial Brook. And Centennial Brook is the one that ran through the city. So what you find for Centennial Brook is that the most common species there represents maybe about 70% of the collections. But as you move further up to the total of 10 species, the abundance curve is very, very steep. Most of these things are still pretty common, and the least common thing is around 0.5%. Now you compare that to, say, both diverse communities like Snipe Island, which has 40 species, and its curve doesn't reach nearly the peaks of abundance of the polluted one. The most common species is only about 20%, and it's a shallower curve, meaning that you're fitting more species and more relatively common species, as time goes by. So rank abundance curves are a quick way to capture the relative rarity and commonness of species. At the same time, capture the number of species you can find in an area. And as we'll explore with our own data, these tell us something potentially about how those communities are put together.